All right. So welcome everybody. I'd like to welcome you to the Research Journal in Medical and Health Sciences webinar. And we are super excited to have Dr. Ebrahim Mostafavi, who will give us a presentation today. The title of his talk will be Design and Development of 3D in Vitro Models in Tissue Engineering and Regenerative Medicine. And I am Tom Webster, the editor of the Research Journal of Medical and Health Sciences webinar. So Ebby, before you start, I do have some announcements I'd like to make about the journal for those of you that might be new to this journal. I'd like to introduce you to it. It's a very broad journal. As you can see by the title, we are interested in publishing anything in medical and health sciences. We have been accepting papers since last fall. And there's our website if you want to go there. We have our articles located free of charge right now for open access. You don't have to have a subscription. You can download those papers. You can also see our webinars. This webinar will be available shortly after it is given. We do have a hybrid. Eventually, we'll have a hybrid mode where we'll have a publication fee and we'll have a subscription in which you can download those articles. We also on our website have article metrics. So it shows you how many people have viewed your paper, which I think is a very nice feature, updated live as people read your work. And of course, we're looking for rapid publication. As we all know, in the health sciences and medical area, keeping up to date on research is critically important. So here's what we commit to do for the journal is publish high quality research, have an open access format to ensure your paper is read quickly after you publish your work, inform you on metrics on your paper. We also are introducing a newsletter that's coming up in which we'll publicize the work that is published in our journal. And as we mentioned, webinars and other types of things are coming up. For example, we're planning on doing a tutorial session for some experimental lab protocols that you might find interesting. So pay attention as the spring into summer moves on, and we'll have some of those nice features to support our researchers and authors. We obviously view publication as more than just publishing papers, but really establishing a network of researchers. I also want to introduce you to a couple of great things coming up. We've established some new awards. So if you're below the age of 45, then you qualify for our Young Research Award. If you're above 45, our Distinguished Research Award. And all you have to do, you can nominate yourself, you can nominate a colleague, and submit the CV and nomination letter to me at that email address that's located there by April 15th. And then we're having a conference. So we're going to have a conference in Kenya to talk about the world coming together to solve health disparities. I think, as we all know, COVID highlighted a lot of health disparities around the world. So we really want to have a conference in Kenya to discuss this. So pay attention to that. It'll be June 12th to 13th this summer. We'll introduce these awards and we'll have a lot of keynote speakers, other kind of contributed speakers. So go to our website and again, keep up to date on that information. Okay, so Ebby, we finally get to you. So I'd like to introduce Ebrahim Mostafavi. He's a postdoctoral researcher at Stanford Cardiovascular School of Medicine in Stanford, of course. So Ebby got his PhD in chemical engineering in 2020 from Northeastern University in my research lab. He got his master's and bachelor's in material science and engineering from the University of Tehran. He's the current associate editor of the International Journal of Nanomedicine. He's also an associate editor of the Frontiers in Nanotechnology, Biomedical Nanotechnology, published by Frontiers. And amazingly, he serves as the academic editor of Innovation in Cell Press, serves on the editorial board of 17 other journals. So I think it's clear that Ebby has his finger on the pulse of research. That's for sure. He's authored over 6 books, 22 book chapters, has an H index of 32, and Abby, you might want to update us on these numbers. They probably have
Mhm.
gone up since we
Yeah,
last talked. He's
these are
also
these are too old.
yeah, so constantly publishing Ebby is so he's also received numerous awards, including the T32 NIH Postdoctoral Training Fellowship. He's also a fellow of the Society of Brain Mapping and Therapeutics. So I could go on and on talking about what a wonderful person and researcher Ebby is, but I think it's best if we hear directly from him. So Abby, I'm going to make you the host and feel free to start your presentation whenever you like.
Sure. Thank you so much, Tom, for the great introduction, and I appreciate inviting me for this webinar. So that introduction, I don't even remember where I uploaded that introduction because that's kind of like too old. Okay, so let me share my screen. Um, okay. Can you see my screen now?
Yes,
Okay, so maybe I just swap. So is it in full mode, full screen mode?
now it is. Yes, it looks perfect.
Okay, great, great. So let me start like uh, today I'm going to present on the design and development of 3D in vitro models, especially for tissue engineering and regenerative medicine. This is definitely one of the fields that I'm very interested in, and I'll be try to very quick and present on the mindset that we have on this. So just a brief introduction. By background, I'm a material scientist. For bachelor, master, I was in University of Tehran, and then I moved to US. I started my PhD at Northeastern, and in parallel, I was working at the visiting researcher at Harvard Medical School. And then 2020, I defended my PhD and started my postdoc at Stanford using human-induced pluripotent stem cells for disease modeling, drug screening. Um, Today, as I mentioned, before going to the main topic of today's talk, I would like to clarify what is kind of my mindset and what is the focus of my research. Here you can see a detailed fellow chart of the research strategies that I'm interested in. My mindset is to combine the fundamental research on the discovery of the different advanced and functional nanomaterial, as you can see here. These nanomaterials are biocompatible and biodegradable, could be eco-friendly and adaptive nanobiomaterials, and it can be a different range of different type of nanomaterials. For example, it can be a stimuli responsive nanocarriers, it can be naturally drive biopolymer, or green synthesized nanoparticle, or functionalized or conjugated nanoparticles, or even like metal organic frameworks. Sometimes, for example, we use MOF or nanosomes for drug delivery. But the thing is that these nanobiomaterials can be implemented alone for later studies, for later investigation, or it can be used in a combination with a 3D in vitro model, which is kind of my interest in combining these two together. And these 3D in vitro models can be a hydrogel, as you can see here. It can be a hydrogel, it can be a nanofibrous scaffolds, 3D or 4D printed constructs, microfluidic system, or organoids, spheroids. But the overall goal is to combine these two approaches and then create some kind of biologically complex systems that can recapitulate the cells, the tissue, the organs, and then investigate, understand or manage different human healthcare, human diseases. My focus is mostly on 4 different applications, which is tissue engineering, regenerative medicine, that I'm going to talk and touch upon it today. It can be like on infectious diseases, for example, on COVID, or on, for example, the inflammation of heart muscle, like myocarditis. It also can be related to some like cancer therapy, cancer diagnostic, therapeutic, especially with focus on women's cancer, breast cancer therapy, and at last on biosensor and bioelectronic for like especially for the cancer diagnostic. Uh, with that, I would like to show you, for example, one of the examples on like cardiovascular regenerative medicine. So as you know, cardiovascular diseases are the most leading cause worldwide. And like, it kind of imposing a very uh, uh, big like cost and like burden on different societies, like not only in the United States but worldwide. And definitely, like for example, heart attack is one of the most like important type of like cardiovascular diseases, and it kind of like it's a death of the cardiac muscle, and then the regional contractile dysfunction, and this can lead to a heart like a death of heart muscle, which is the heart attack. We call it like myocardial infarction. But tissue engineering and regenerative medicine for sure can bring some like promises to solve the issue with this like a heart attack or myocardial infarction. But as you know, cardiac tissue is the most complicated tissue in the body. It not only contains these like very highly aligned cardiac cells, it's also contractile, it's conductive, and like the community, they're trying to mimic, recapitulate these complex structure, and there was very good like progress from different researchers worldwide that could successfully recapitulate these kind of complex structure. But the other obstacle that hinder kind of the translational application of tissue engineering regenerative medicine approach in cardiovascular regenerative medicine is the lack of effective vascularization or like angiogenesis. But with the implementation of these bioengineered scaffolds, we are trying to overcome these issues. As I mentioned, heart attack, there are so many like different approaches that can be carried out for like this heart attack, myocardial infarction. Heart transplantation definitely effective, but we have a limited availability. Cell-based therapy, there are so many good studies, very great progress during the past 2-3 decades, but still there are so many like obstacles. As you can see here, the viability and phenotype stability of the cells are very low. The ineffective homing of cells, or regulatory challenges, or exorbitant cost. But we hope that bioengineered cardiac scaffolds can solve these problems. But these bioengineered scaffolds, as I mentioned, these could be in different types. Like it can be in the shape of a cardiac patch, which is a layer of the like cardiac functionalized cardiac tissue that can mimic the native myocardium. It can be a injectable scaffolds or injectable hydrogel, or it can be a 3D or 4D printed construct. But fortunately, I was lucky enough to work towards all these kind of like bioengineered scaffolds. But today I'm gonna show you a few of these scaffolds that we developed in the lab during the past few years. For example, as I mentioned, this figure shows the various type of 3D in vitro models that can be used as electroconductive scaffolds for cardiac tissue regeneration. For example, this can be a patch, it can be a hydrogel, it can be a 3D bioprinted structure, or it can be even a combinatorial approach, like 3D bioprinting. That I also show you one slides about like how we used 3D bioprinting with electrospinning techniques to make such kind of tissues, and it can be also injectable hydrogel. But what I always try, as I mentioned, like by background, I'm a material scientist. I try to be very cross-functional in my research and have like multidisciplinary approaches in the project. So I always try to kind of combine nanomaterials with biology and biomaterials to make these kind of nanostructured, functionalized and advanced nanostructures for different disease. Here for cardiac myocardium regeneration, or for heart repair. So the thing is that, like as I mentioned, for cardiac tissue, you have to have a structure that is not only aligned, it's also conductive, it's beating, like the conductivity is very important. But the non-conductivity of the extracellular matrix limits the application of the polymer for the modulation of the excitable cell types, for example, cardiomyocytes. But here, we incorporate these nanospecies to extracellular matrix to provide this kind of conductivity to the scaffolds, and therefore we kind of overcome these limited intracellular connections, and then we can provide this electrical signal propagation. So here, this is just one example that they use, gold nano wires. But during the past few years, I tried different nanospecies to provide these conductivity to the scaffolds. For example, here you can see some of these nanospecies that I personally use, like graphene oxide, reduced graphene oxides, graphene nanoribbons, and even multiple carbon nanotubes that was functionalized. But today I'm going to show you some data that I use the graphene nanoribbons to mimic these kind of conductive structure. And here you can see a flow chart of the whole idea, that first we have a gelatin-based material. It could be gelatin. Sometimes, based on the needs, we also metacolate this gelatin to make the gelatin metacril oil, and then incorporate these nanospecies here, graphene nanoribbons, and then using a technique here, we use electrospinning. But as I said, this could be different approach. But here we use the electrospinning to make these cardiac patches, and then first and foremost, as a bioengineer, I always try to optimize the condition for mimicking the tissue that is the target tissue. For example, here cardiac tissue. So first and foremost, definitely we need to look into the physical chemical property of that structure, and to make sure that the structure well enough recapitulate the native myocardium in this example. And then when we have come up with this optimized condition, we can definitely in later stages, we can definitely functionalize this biologically with different type of cells. We use back in my PhD, I used neonatal rat to isolate the cells from 2-day neonatal rat, and then isolated to cardiomyocytes and cardiac fibroblasts, and then seed these cells on the scaffolds, and then later it can be implanted in vivo. So here I'm just showing a very brief a slide on the data. So the thing is that like when you incorporate the graphene nanoribbons compared to the pristine extracellular matrix, which is gelatin, we could see a very well developed and interconnected sarcomeric alpha actinin, which is an indication of the like the uniformity of the like structure, the cytoplasm. And then also you can see that by incorporating these graphene nanoribbons, you can see a very uniform gap junction, which is obvious from the overexpression of the connexin 43 on the surface of the graphene and gelatin patches. But the thing is that, like, interestingly, I also try to incorporate some of the epicardial secreted factors to these patches and see how these epicardial secreted factors can kind of like affect the regeneration of the myocardium. And the interesting point was that when we use the epicardial secreted factors protein, we can see a very significant difference in beating rate of the cardiac patches. Even these epicardial secreted factors, the mechanisms was kind of like developed in the year 2013 or 2014 in a Nature paper, and they also showed these epicardial secreted factors not only increase the number of the blood vessels, which is the angiogenesis, and increase the vascularization. It also accelerate the division and proliferation of these cells. And when we have the folloestatin like from as an epicardial secreted factor, we can see that we develop these very nice cytoplasm, and the number of the cells are increasing, and also the maturation index, for example, is increasing. The thing is that, like, since we are working with carbon-based material, we definitely need to be aware of the the cytotoxicity, because that's why I always emphasize that we need to come up with the best condition. Like, for example, for these graphene nanoribbons, the concentration that I could use to get the minimum toxicity to the normal cells, to cardiac cells, was, for example, 0.1, which you can see here. But for example, for carbon nanotubes, it was different. For like graphene oxides, it was another threshold. But anyway, we are not looking for some scaffolds that provide the best conductivity, because we are going to optimize this conductivity based on the native myocardium, and we are not looking for some scaffolds that are beating, for example, 5 times more than normal cells or normal cardiac tissue. But interestingly, so here you can see, just the 2 video that shows when I incorporated the epicardial secreted factor into the patches. You can see some very synchronous beating throughout the whole structure. Maybe this is not clear enough to show, because the beating was very thorough and very synchronous beating. But here, I took a video with my iPhone, and you can see as you can see, the whole patch after 10 days of the incubation is beating as a synchronous structure, and this was very, very promising, and it was I would say 70 to 80% reproducible, which was a good point. And then one of the interesting point in that and this research was that most of the research that I see on the literature, they report 12% like contractility. But the thing is that like they always use some external stimuli, like mechanical stimulation or electrical stimulation to give that contractility, 12%, for example, contractility to the engineered tissue. But here we use only the protein epicardial secreted factors, and we could reach to almost 11% of contractility, which was very promising. And so, but the other point was, as I mentioned, like we use carbon-based material to make these cardiac tissue, but carbon-based materials can provide these antibacterial activity, make an antimicrobial material. And as I mentioned, one of my interests is definitely antimicrobial and antibacterial, antiviral research. And for that, I wanted to look further and see if these patches that I engineered can also help treat the inflammation of the heart muscle, which is myocarditis. And this myocarditis is usually caused by Staphylococcus aureus. So I tried to characterize or investigate into the effect of these patches in killing or preventing the bacteria to grow on the scaffolds. And so the reason for this is that the emergence of the antibacterial resistant bacteria is something that definitely the antibiotic overuse because of the bacterial resistance is kind of causing a very health crisis. For example, the approval of new antibiotic by FDA are really declining, and within the next decades, we definitely wouldn't be able to see any approval on these antibiotics. So definitely we need alternative strategies, like using, for example, nano systems, nano platforms, or some kind of drugs. And when I characterize these scaffolds, they engineered the scaffolds with graphene and compared that with a commercialized microbial antibacterial material as chitosan, I could see a very similar behavior. And for example, here bacterial colonization of the surface of these cardio patches shows that when we have only the extracellular matrix, the gelatin, the bacteria, both gram-negative and gram-positives, are growing very well. But in terms of my engineered patches and also the one that is commercialized with using the commercialized version of chitosan, you can see both showing the same kind of behavior. And the live data assay also shows that after 24 hours there are so many dead bacteria over there. But this was just a cardiac patch. And as I mentioned, today's focus is just 3D in vitro models, different 3D in vitro models. For example, we can use these 3D in vitro models for other kind of tissue engineering. For example, we use these for in vitro models for wound healing or skin regeneration. For example, we in one study that was published in European Polymer Journal, we developed this chitosan with PVA hydrogel, and then we incorporated the green synthesized cerium oxide nanoparticle to see the application of the to see the potential for wound healing application. Here you can see just some cross-section SCM images of the hydrogel with 3 different concentration of the cerium oxide nanoparticle, and then human dermal fibroblast studies also on cytocompatibility study shows they have various hydrogels over 5 days of the culture, and you can see the metabolic activity of the cells are significantly increasing over 5 days of culture. Or in another example, for example, we can use the same approach, for example, electrospinning, to make tissue engineered vascular grafts that are perfusable, that are anastomizable, and you can later use these as a like plain artificial blood vessel that can be implanted in the, like, you know, for like some type of, like, you know, suturing. Or it can be like, the purpose of my study was to incorporate these artificial blood vessel in, we combine it with a 3D bioprinting and see how we can generate a like thick tissue. But here you can see a schematic that like we got the gelatin, methacrylate gelatin, gelma, then we incorporate some like, you know, synthetic polymer, and then electrospin it, and then using a bioreactor, peristaltic bioreactor, we could kind of endothelialize this vessel. And then here you can see some characterization of these blood vessels, like, you know, for example, the tensile strength, the mechanical property, and comparing that with the native porcine blood vessels. But the point is that, like, later the kind of like the idea was that using these vessels and then 3D bioprinting a hydrogel that includes the, like, for example, smooth muscle cells, HUVEX, endothelial cell, like, you know, some growth factors, for example, VEGF, around it, and then monitor it for, like, you know, incubate it or like perfuse media or blood into this structure, and see like how this affecting the, like, the vascularization of the tissue. But here you can see just an example of like different methods that you can generate these vessels. What I showed you was the electrospinning, but I also try to make these, like, you know, blood vessels with 3D bioprinting, or like mold or casting molding tube. But as I mentioned before, the idea was using these vessels and then 3D printing a gelma that has HUVEX, and as you can see here, over 7 days of culture, the cardiac cells in the 3D printed structures are viable and are proliferating very well. So this was another, like, you know, approach, because, you know, as we also we have some like collaboration with Tom, that we working on 3D 4D printing material, and like definitely 3D or 4D printing is definitely one of my interest. Or for example, here in another study that we kind of looked into the synthesis and application of the kind of like glycan nanoparticles, and then incorporated these glycan nanoparticles with laponite to make a shear thin hydrogel, and then 3D bioprinted these hydrogels, and we kind of made these cell laden construct, and then optimized the whole like 3D printing process. For example, here in this, here you can see the stability of the structures over 3 weeks in physiologically relevant environment. And then as you can see, these structured 3D printed structures have a very nice stability, and later we also use these constructs to inject it in a kind of subcutaneous model of the rat, and after 7 and 14 days, we post injection, we looked into the effect of these 3D printed hydrogel, shear thinning hydrogel. Also, in another study was a collaborative study when I was in Tom's lab. So we used like the in situ printing of the scaffolds for bone tissue engineering. For example, we had a, like, you know, handle that could print the structure, and then these structure were incorporated with zinc oxide nanoparticles, and they also gave very nice antibacterial activity. So the whole point, like so far, was like using like primary cells, for example, to develop these 3D in vitro models. But the thing is that like when I was back in my PhD, I was working with, as I said, primary cells, or like isolating cells from neonatal rats, which is a very labor-intensive process. And like, you know, it was such a like pain going through the process. Like every 2 months, I had just one batch of cells to do the experiment. But you know, later I was thinking about, like, after my PhD, what if we use a more like reliable and more kind of like uh, more reliable platform, as like, for example, human induced pluripotent stem cells, because these cells can be generated, like, you know, can be from any, like, you know, individuals, and it kind of like help us to go towards the precision medicine, precision healthcare. You can get the, like, you know, reprogram the cell from any patient. You can have the healthy cells. You can have the like disease cells, and then, you know, differentiate this towards any kind of cell types. Especially, for example, I was working on the kind of you know, cardiac related cells, like cardiomyocytes, fibroblasts, endothelial cells, and then and also the efficiency of those cells are much like, you know, more than like neonatal rats. As you can see here, the cardiac cells differentiated from iPS cells, um, showing, like, you know, beating after 11 days of culture. And then I use these, like, you know, human induced pluripotent stem cells to kind of make the cardiac organoids. These cardiac organoids, as there are so many different, like, you know, methods to make the organoids, like cardiac organoids. It can be just by like differentiating cells and then mixing these, or and fusing the cells to make these organoids. But the one of the downsides of these organoids is that, like, you know, you cannot use these for like disease development studies. Definitely you can use it for disease modeling, for like drug evaluation, for transplantation. But there are different methods. Like, for example, you can aggregate these human-induced pluripotent stem cells and make the organoids. But the point is that, like, for example, here I'm showing, like, you know, one of the organoids that embedded in a matrigel as an extracellular matrix. You can see the beating of the organoids as a, like, you know, in day 22 after culture. And for example, one of the like applications that I'm interested in is to use these cardiac organoids for, like, you know, drug screening or for disease modeling. For example, even we can have billions of these cells, these organoids, and then jam it in a, like, you know, in a syringe and then 3D print it. So okay. So this was the end of the presentation, and at the end I would like to acknowledge the support, and, like, you know, I would like to thank the different journals that trusted me and have me on their board as associate editor, like academic editor, or, like, you know, editorial board member. And also I would like to acknowledge all the agencies, like institutions, labs that supported my research during the past few years, especially like when I was a PhD in Tom's lab. I could do a lot of research. And so with that, I would like to take any question. This is a beating cardiac organoids, which is like, you know, kind of like shaped cardiac organoids. So I would be happy to answer any question.
Excellent. Thank you, Ebby. That was great. I love
Thank you.
the thumbs up. So we have time for some questions for Ebby if anybody has a question. I can certainly start, Ebby, with a question. So this
Mhm.
is more of a futuristic thinking question. And I know we're both proponents of 4D printing, right?
Mhm.
Where you can control remotely the shape of a material over time, right? And a lot of your research really seems like it's going in that direction, right? Where you used graphene nano ribbons or nano rods or nanoparticles. Some of the materials had gold nanoparticles. So all of those can heat up, right, under near infrared and cause this four-dimensional time change properties after you implant a material. But it always seems like in 4D printing, the weakness is the material that you add, right? You know, the toxicity of
Mhm.
graphene, right? Or the toxicity of gold nanoparticles. I'm wondering your thoughts on that. You know, do you think what is keeping 4D printing from going forward is having a biodegradable, safe material that you could add to Gelma
Mhm.
or to polymers, you know, because at least what I'm seeing is there's a lot of concern, you know, from the FDA, from people, that material that gives it a 4D property is also its weakness in terms of possible toxicity or permanency, non-biodegradability.
Yes, that's actually a great point. And as you mentioned, like, there are so many doubts about, like, using these 4D printed structures because of that toxicity problem. But I would say, like, there are so many good progress on that. Like, like, for example, one of the things, I mean, I never worked on this directly, but, one of the things is that, like, instead of adding, like, nano species to give that kind of, like, property, you can use some, like, naturally, like, for example, biopolymer that gives that, like, for example, property. For example, in this, like, case, for, let's say, for cardiac, some people using the naturally, like, biopolymers that have that are conductive, like polyaniline. Like, this is something that definitely maybe in the future. I still, like, as I said, I never worked on those, like, myself, but I believe, like, this is some, like, avenue that definitely researchers can look into that, like, but still, since I was always working with nano species, I still believe that there are so many rooms to work uh, towards, like, making a translational, like, 3D or 4D printed structure that is incorporated with nano species. Like, one problem is, or, like, especially when you talk to a clinicians, like, they do not trust very well to nano, anything nano, because,
Yeah.
they feel that, like, this is just a fancy stuff. But still, we as nanotechnologists, I believe there are so many ways that we can improve these until we get to a translational, like, application of these 3D or 4D printed structures.
That is a great comment, you know, about polyaniline and whether to use polymers as that material that responds to near infrared rather than these metal or metal oxides or nanoparticles, you know, and maybe a forward thinking idea too is I love your organoid approach. And obviously you're using cells to cause that beating, but perhaps in vitro assays might maybe there's an advantage to not using cells and using a polymer in an organoid to beat without cells, you know, where maybe you want to separate the effect of the cells in terms of some drug metabolism or something like that, and you have a construct that's acellular, but it can beat
Mhm.
at the same rate. You know, maybe that's a home for 4D printing where you don't have to be concerned about regulatory approval because
Yeah.
you're using it as an in vitro assay. I don't know.
Yeah. No, definitely, this is a great idea.
Other questions for Ebby?
It's not a question. Maybe I can add on something in the same line. Organ or no, organoid is a very great concept, and I think we have to pay more attention to this field.
Mhm.
As we know, this is tough to create a microenvironment which is good for health using technology, but it's possible.
Yeah, yeah,
Yes.
exactly. Like, I'm
And
kind of like Ajit, I'm kind of like, maybe like it's been 2 years that I got interested in organoids, and I believe organoids is such an amazing platform to like look into different diseases, or like, as I
yes,
mentioned,
yes,
you can use that for like, to study the developmental stage of a like, disease. Like, for example, we use it for like to see how the congenital heart disease is happening, like, what are the stages, or like, you can also use that. Maybe I would say like the less promising aspects is still like, it's very challenging, is the translational.
yes, yes.
In having a like a structure that can be implanted later, like, for example, for cardiac myocardium, injury.
Yeah,
So,
but
but
I
the
don't
other
know.
step,
You're right. I don't know why people don't take it as an alternative of existing techniques, which are very expensive and needs a lot of animals to get
Well,
sacrificed.
yes,
Yes,
or
and a lot of yes, 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 a lot of work, a lot of in
yeah,
vitro, a lot of in vivo investment. This can be replaced by organoid or can be reduced. Like rather than
yes, but
compromising 1,000 animals, someone can optimize a lot of things
yeah.
by organoid approach and optimize the same thing using 100 animals.
Yeah, but one thing, I assume that maybe you never worked like, with organoids in your lab, because if you work with organoids, you would be like surprised how difficult is that to work with
Yes, it is.
organoids, and like it's very, very difficult. The cell culture, the usual cell culture that you use, like for example, for different kind of like 2D, 3D cell culture, is just a simple task. But when using like organoids, the task is very, very challenging. Handling these
Yes.
organoids, like even when I like, I'm always thinking about like developing these 3D, 3D printed dishes that can hold organoids in place, and then you can like, start culturing, because when you pipette these like, organoids, you would lose like 50% of the organoids in one culture, and you don't know, because these are tiny like guys that, with every pipette you might lose them. So
Yes.
but there are so many companies, like, I had some like meeting with a CEO of some companies in Bay Area that, they're working towards not developing the organoids in a like consistent and real like reproducible manner, but they're working towards making these organoids in a way that they can like downstream assays. They make it the downstream assays very easy, but that's not what I'm interested in. So I always think about like, one step before that, like, how to make these organoids a reproducible platform, like, that you can, for example, in one run you can have like, hundreds of these organoids, then, add different drugs and see, like, because, these organoids is the best platform, because when you have like the cells, for example, let's say you have my cells, I have a disease, and then you can recapitulate like, the same body, my body, in the dish, and then add different drugs, and then that's why we are getting closer to like, to personalized medicine with these organoids. But still, there are so many things that need to be like, addressed in using organoids.
Or agree.
and I think a wonderful future avenue of research, and perhaps this is already going on, but of course, Ajit and Atit, you guys know a lot about sensors, and organoids can respond much better to chemical anomalies, right? Or
This
to,
is
so
true.
can you combine organoids into the sensors that you develop, that
The
not only
problem
for,
is
yeah, so
it's a
not only
very
for
yeah.
medicine, but maybe for non-medical applications, to look at various chemical analytes that might be contaminating the air in which those cells, respond or die or swell in a much easier way than individual molecules, right? We always tend to think about biosensors as putting an antibody or putting, you know, a functional group, but the organoid could be much more sensitive, right, to those changes in chemistries that if they could be combined in a biochip, you know, maybe that's a better sensor.
I agree with that, because at every process, cells has to respond, and they will release many chemicals, many markers, kinokine. I'm not good in biology, cytokine, chemokine, because back in 5 years ago, I grew cells and infect them with HIV, and I treated the same cell using anti-HIV drug, and I grew that cell on electrode, and I checked their impedance, and every time I see the change in impedance, and I got US patent, and I published a paper on that. I wrote a grant to NIH. They did not believe me. I have to quit that area because no money is.
Yeah.
That's the sad part of academia.
Yeah. Absolutely. But you know, it does really, I know it's more complicated to put an organoid on a biosensor, but
I think we need industry. We need you for that. Forget NIH. We have our own business.
But if you think about it, right, and Abby, you would appreciate this, that we spend so much time trying to interpret whether one specific chemical increases in the body and what that could mean for a heart attack. But wouldn't it be much easier just to have an organoid, a heart organoid on a chip, and see if that chemical causes that to have a heart attack? You know, then you don't have to interpret. You're looking at the chip and you're seeing indications of a heart attack. So you don't have to interpret that individual chemical because you have your heart organoid that's having a heart attack. So you know that that's a problem. But you're right, Ajit. You know, the industry has to come apart. And Evie, I thought when you mentioned Silicon Valley, maybe some of those companies are looking at combining sensors with organoids, but I don't see a lot of that activity right now.
And my idea was that if we can do that, we can replace even ELISA-based testing. We can replace it with PCR-based sensing. We don't need these techniques, but have a one-time correlation, response, magnitude of electrical response with reference to the parameter we are looking for, and then just go for it.
Yeah.
Yeah, actually, I would like to add something. Sorry, Abby, if you want to continue, please go ahead.
You go ahead. No, you go ahead.
Okay, thank you. Thank you, Abby. Actually, it was a very nice presentation. And being not from biological background, even I learned a lot actually today. Thanks a lot. And Dr. Thomas, actually, like you told, that's a very good point, because just I was looking, I just started Googling at this moment, and it is showing that there are some reports, very recent reports, organ on a chip platform for heart disease and all the things Abby mentioned with zinc oxide nanoparticles. So if we can design something with zinc oxide nanowires, for example, they are much more sensitive, higher surface area. We can design on a chip kind of device with nanowires, for example. Zinc oxide, they are so much sensitive to any change, like chemical change, electrochemical change, piezoelectric change, electrical change, optical change, all those kind of things. We have to see what kind of parameters we can go for. And I think we can design something basic device prototype in the beginning stage, like Dr. Ajit was mentioning. Of course, it won't be as sensitive as compared to the ELISA or those kind of conventional techniques, but at least could be the first step, something basic for a large population. For example, we can do a general test, and out of that population, we can send those special people who have given some kind of positive response. They can do ELISA now afterwards. So I think we can suggest that kind of sensing device or something. Yes, that's my idea, I think.
Excellent. It sounds like the foundation for a company.
Yeah.
and then
like Dr.
I was going to say,
go ahead.
and Greg, you could make the zinc particles using your laser ablation.
Oh, I didn't know that Dr. Gray also, he worked with zinc oxide nanoparticles.
But yes, lots. Sorry, go ahead.
Maybe I have a quick question.
Sure.
You are using, your zinc oxide nanoparticles. Is it, for their antibacterial properties or because they are piezoelectric?
No, for the antibacterial. Like in that specific like case that we published in Acta Biomaterial, yeah. So it was for antibacterial. Especially we are interested, we were interested and still interested, and we have some collaboration with Yogendra that we are interested in using those tetrapods nano particles, zinc oxide tetrapods that Yogendra developed in his lab.
Excellent. Well, I'm looking at the clock and it looks like we've used up our hour, but let's thank Ebi. That was absolutely wonderful.
Thank you.
Presentation and thought-provoking, especially at this interface of science and organoids and 4D printing and so much. So as a reminder to everybody, please submit papers to the Research Journal in Medical and Health Sciences. Go to the website, keep an eye on our conference coming up and our future webinars. So thank you all very much. Thank you, Ebi, especially, and we'll see you guys soon.
Thank you.
Thank you.
Bye.
Bye-bye.
Nice meeting you, Dr. Gregory. Thank you, Avi. Thank you, Dr.
Yeah,
Kaushik.
sure.